much for coming out, and I hope you've really been enjoying the different demonstrations and having a chance to chat with people about the different research projects we do here. My name's Matt Lamban-Rife. I'm the director of the unit here, and so it's always lovely to have people come and visit and see and get to know a little bit more about what we do and meet some of our fantastic researchers in the unit. So what we have for you now is 4 short talks with a chance to ask questions if you've got any in between, and it covers gives you a little kind of sampling of some of the things we do in the unit across different research areas. We just wanted to give you a taster of some of the different projects and some of the questions that people think about in their research. So we're going to start with Lexi, who's going to tell us about simulating brains and how it works and what we learn by doing so. So over to you.
Thank you so much for the introduction. I'm really excited to talk to you all today about brain simulations. In neuroimaging research, we often break up the brain into regions or areas. However, we also know that the connections in the brain are important. So if you think about areas of the brains as locations on a map, the paths that connect those areas hold a lot of information about how the brain functions and develops. And thanks to a very simple property of water, we can actually map the connections in people's brains. So we know that if water molecules are in an open space, those molecules will move around freely in any direction. However, if there's some type of barrier present, the molecules would take the path of least resistance, and so they'll move along the unrestricted direction. And so if we think about what a simple neural structure looks like, we know that the neural projection, so where the signal is being moved through space, is surrounded by a tissue called myelin. And so if we actually just cut this neuron in half, we would see that the myelin surrounds the connection, essentially making it into a tube. And so if water is present inside those connections, myelin acts as that barrier, which means in your brain, water molecules are going to move along the direction of the connections. And so we can actually picture the direction of the movement of water in your brain using magnetic resonance imaging, or an MRI, which gives us a scan which captures which way the water molecules are moving. And so we can actually trace this movement and create a 3-dimensional reconstruction of the connections in a person's brain. These are called connectomes, and they hold a lot of very detailed information of all the connections that we've picked up on a scan. And so in order to analyze this, we often break this down even further by combining it with what we know about brain areas. So this is an example of a brain network where the circles are depicting areas of the brain, and the connections between those areas are being depicted by the lines between the circles. And so we would call this a macro-scale connectome, which means we're not looking at connections at the basis of a neuron-to-neuron connection. We're looking way more broadly than that at how areas of the brain are connected. And we can think about these networks the same way we'd think about a road map of how roads are connecting different areas of a city. And so this is simply a 2-dimensional version of what I was just showing you. But when we create these networks, we have the opportunity to look at how the whole brain is organized. So if we zoom in on a small part of this network, we can calculate things such as the number of connections that an area has, how clustered different areas are together, and we also can calculate efficiency. And this is a really important concept because in the brain, distance is considered costly. And this means that if a physical connection is going to form between 2 areas of the brain that are far apart, it's going to take the cells more time and energy to form that connection than if the connection was much smaller or closer together. However, it's also important that the brain is well connected. So efficiency is essentially a trade-off between these 2 things, being well connected but also minimizing unnecessary cost. So you see here that 2 regions are labeled A and B. There's many different ways that A and B could be connected, but the red line is indicating what would be the most efficient connection because it's the most direct in minimizing unnecessary costs. And so researchers have been exploring how organization of the brain relates to important outcomes. And so from some research that was conducted here at the CBU, people have found that in children, those who display higher efficiency networks compared to their peers also typically display higher cognitive abilities, higher general intelligence, and better academic attainment. And so from this kind of research, we know that brain organization is important and it's related to cognitive and behavioral outcomes. And we also know that people's brains are organized differently. We see naturally that some people's brains are more efficient than others. And so the next logical question, or at least logical in my mind, is how does brain organization develop? And so what I mean by organizational development is how does the brain go from having, say, 50 connections to having 400 connections? And most importantly, how and why are those connections being the ones to be added? So why are some connections made and some aren't? You could think about it the same way you could think about a city. If you have a new city with a few roads, how and why would you choose to make roads that connect different parts of that city? And this is where simulations become very helpful. So these are connectomes of infants, and as you can see visually, the amount of connections being added in these infants is rapid on the scale of just weeks. And so to conduct this research, we would scan an infant. Let's say we scan an infant at 35 weeks, and we can create their brain network from that. And maybe we're lucky and we actually scan the same individual again 10 weeks later. And so from this kind of data, we have two static points in time where we can look at and measure the type of organization that that infant has at 35 weeks and 45 weeks. But from neuroimaging data, we don't have a way of learning why or how was that organization changing from 35 to 45 weeks. We can only see what the changes were. However, we can do something called a generative simulation, generative simply indicating that it's growing, where we grow essentially a fake network over time. And we can match a simulation with an individual. So these observed networks is what an actual individual's brain would look like. And we pick a simulation that is similarly organized. So it has a similar level of efficiency or clustering and a variety of different measures. And the benefit of doing this is we know how these models were choosing to add connections as they were developing. And so it gets us one step beyond what we can just observe by neuroimaging work and a little bit closer to understanding the multiple different paths that can lead to the same endpoint of being an organized brain. And so now I'm going to get into how these models actually work. So in this research, we give the model all the regions of the brain and the distance between those areas, so depicted by the circles here. And oftentimes, researchers will include a few connections that are present in the vast majority of people. So these red lines might be indicating connections that are present in 95% of the people that we're looking at. So what the model is doing is it's adding connections. It will take this data that we provide it, and it will add connections until it has the same number of connections as the person that we're trying to simulate. So what is important about these models is not necessarily what it's doing, but how it's doing it, how this model is choosing which connection to add next. And that's based on this equation. So P is the probability of two regions connecting for this small example A and B, and that's determined by cost times value for that connection. Cost, as I've been saying, is distance. So these models are going to preferentially pick connections between regions that are closer together. And value is having similar connections. So as you can see, A and B share a connection with a third neighboring area. And that means A and B are more likely to form a connection with each other than they would be if they didn't share this connection in common. And so what we do is we create tens of thousands of these simulations that weigh cost and value slightly differently. Here I'm just showing you 3 examples. And they weigh cost and value by being raised to a number. Cost is always negative because we're penalizing costly connections, and value is always positive in weighing how important valuable connections are for that model. And so what we end up with are models that have slightly different conditions. The first model is driven mostly by cost. It's raised to -4, which in this case is highly penalizing cost. Model 2 is a bit more balanced, whereas Model 3 is driven mostly by value because it's raised to 0.9, which in this context is weighing value very highly. And so if we look at how this looks throughout the generative process, we have 2 examples here of the most extreme. So on the left, you have a model that's simply trying to minimize costly connections. On the right, you have a model that's trying to select connections that are valuable. And what we see is as these models develop, they're going to organize very differently one with lots of short-range connections, one with lots of long-range connections. And we don't see either of these two scenarios in reality. People's actual network is a trade-off, or a wiring trade-off between cost and value. So people's brains appear to be organizing in a way that does minimize cost, but also allows for some longer-range connections to form that are valuable. And what's really interesting in a lot of the work that's being conducted with this type of simulations is that we see individual differences in people's wiring trade-offs. And so I'm going to summarize quickly a few different ways that this has been applied in the past. In my first year as a PhD student, I was looking at infant development and found that wiring trade-offs were different for infants that were born premature compared to infants that were born at term. Past researchers also demonstrated that wiring trade-offs change across healthy aging, so older individuals are simulated based on different wiring trade-offs compared to younger individuals. There's also been exploration to cognitive abilities, so wiring trade-offs in children appears to be related to their cognitive performances. And there's a lot of different researchers looking at how this relates to genetics and even how wiring trade-offs and genetics relates back to cognition. And so I'm going to summarize two big takeaways if these are the only two things you listen to me about today. First, brain networks can be simulated by cost and value trade-off. And secondly, this wiring trade-off is related to important outcomes. And what's really interesting about this kind of work is this type of simulations is relatively new. So there's a lot of work still being conducted to learn more about what type of information can we be drawing from these simulations. And I just want to thank my supervisor, Duncan Astle, and my lab, and point you towards this paper performed by Petra Vertes in the psychiatry department here. This outlines the model that I was talking about today if you're interested in learning more. Thank you so much.
Fab. So any questions for Lexi? Yes.
Sorry if I missed it, but could you explain your concept of value?
Yes, so value, there's many different things we could put in for value. So this is something that researchers have already explored a lot in the past. And the kind of consensus appears to be that it's having similar connections. So regions value other regions that share connections. So essentially, if you think about it as share with the node that it's determining to connect with. So for example, if me and you were going to make a connection, if we shared a friend, we would be more likely to connect. It's essentially the same concept of friendships in that way, of if you share a connection with other regions, it's more likely to form that connection.
Yes.
Is there a difference between people behaving for who have more cost-beneficial or value-driven brains?
Behavior hasn't really been explored yet. So it's I think the closest would be kind of the cognitive performance stuff that people have looked at recently. But I don't know of any papers looking at it related to behavioral performance. It would be very interesting to explore that, though.
Yeah.
Is there any indication whether the value connections versus the cost-efficient connections are informed more by genetics or by, say, environment or something like that?
I don't think there's been much exploration of the environment, but there has been some exploration of genetics. And so there's some genetic atlases where essentially you can look at where different genes are being expressed more in the brain. And there has been past research to explore that and demonstrates that essentially when there's a high value, it relates to genes that are related to that process, whereas if it's high cost, it's related to genes being present to like the formation of cellular connections. So there is a paper I can point you towards if you're interested in learning more about that. That's not my work, but there's definitely been some work into that. Yeah.
Yes.
Are there any examples where one sort of connectivity or favour has been shown to favour one sort of cognitive task and a different sort of connectivity favours a different cognitive task?
Not with these kind of models. So these models that I was talking about today don't perform. It's very structural, so it doesn't relate to kind of any cognitive performance. There are some researchers starting to bridge that gap with actually these exact models, but has yet to be demonstrated.
Very good. Shall we move on to the next one? Thank you so much. That was great. So great. So next is Rachel. So Rachel is going to tell us about some mental health research in adolescence.
I am when I find where my slides
Yes.
are.
Yeah. We're here in.
Is Vicky here? Hold on.
Okay.
Sorry, I'm not exactly sure. Oh, okay. Oh, perfect. Okay, yeah, there we go.
Yeah.
All right, sorry.
Simple solution. Great.
I love a simple solution, honestly.
It's fine. I think it's coming.
Okay, it's all good.
Sorry, Vicky.
Perfect.
Yeah.
All right, sorry about that. So yeah.
Great. Over to you, Rachel.
So yeah, my name's Rachel Knight, and I'm a third-year researcher, PhD student in the Cognition, Emotion, and Mental Health Lab. So I'm going to be taking you on a slightly different tangent of what we do here at the unit. So I'm going to tell you about a new low-intensity intervention to improve mental health in adolescents. So I guess a brief introduction to this. Many of you may be aware that adolescence is a key period of development. And this is also the case for mental health. So this is where symptoms generally start to emerge. This makes it a perfect time to intervene in mental health. So before these difficulties become more entrenched and before they start to affect individuals' ability to live a fulfilling life. Now, there have been a number of large-scale studies looking at interventions that we could give to all adolescents, so maybe we could improve everyone's mental health. These are often conducted in classrooms and by trained teachers who have been kind of trained in whatever package of care they're looking at. However, we find that often these aren't very effective. They also tend to be very resource-heavy, both in terms of time and cost. And they're often not seen as very engaging to young people. They find them quite boring. So for our intervention, we decided on a few things. So we decided we wanted to train just one key ingredient of psychological therapy to see if that alone would make a difference to people's mental health. We did that using a digital intervention. We wanted it to be available entirely through podcasts and kind of free platforms. This meant that it would be easy to deliver, scalable, and also we hoped it would be appealing to young people because it was on platforms that they already know about. We also wanted to teach a skill that was specifically practical, so something that if you were having a difficult moment, if maybe you were having a difficult thought or feeling, you could use to maybe reduce your distress. So you're kind of probably wondering at this point, what is this skill that we're teaching? So imagine for a moment that you're about to give a big presentation, maybe at like a public engagement event, you know? So you might feel a bit nervous about that. Disclaimer, I'm not saying that that's a mental health difficulty. I actually think that's quite normal. But you might feel nervous about doing that. So something you might do is maybe you call one of your friends, and maybe you call one of your mates and they say, you know what, maybe you can imagine what it'll be like a month from now. So when you're looking back at giving this talk, will you kind of remember how stressful it was, or will you think, oh, I'm kind of glad that I did that? Will you even really remember giving the talk at all? Maybe you have another friend who encourages you to journal and, have a little look and kind of, write down the feelings that you have and, maybe try and see them from a different perspective. Maybe you talk to another friend and they tell you that, obviously it's normal to have kind of difficult thoughts and feelings about doing things, but that can't actually stop you from doing what you're doing. Even though your thoughts are there, you can still do what you want to do, if that's give that talk. So what do these things have in common? All of these are ways of stepping back and noticing the world from a different perspective, and we call this psychological decentering. So to kind of summarize, decentering is an ability to notice day-to-day difficult kind of thoughts, feelings, memories from a different perspective, often one that's not influenced by personal feelings or opinions, so we call that an objective perspective. And when people do this, it tends to reduce the impact of the distress of these thoughts, so that's why we chose it. So why do we think this is a key ingredient? So one of the reasons that we think that is because it exists across different schools of therapy. So all types of therapy have something that looks like this. So we think it could be maybe one of the reasons that therapy works. Another reason we chose it is because it targets difficult thoughts and feelings, which are something that we know kind of in the clinic and anecdotally are one of the things that's most difficult about having a mental health difficulty. We also know that decentering has a unique relationship with symptoms. So I'm going to show you a graph, and on the y-axis, so the one going up, you've got depression symptoms, and on the x-axis, so the one going along, you've got the decentering. So what you can see on here is that people who rate their depression symptoms as really high tend to rate themselves as quite poor at decentering, whereas individuals who have higher scores on decentering tend to have fewer depression symptoms, and we notice that with anxiety as well. We also know that decentering seems to change over treatment. So if you have someone who's in therapy and over the kind of maybe 8 weeks of therapy, you give them a questionnaire every single session, one that's about decentering and one that's about mental health. What we find is that their decentering scores tend to increase over the course of therapy, and then at a certain point, symptoms start to decrease. So we think maybe it could be that a critical mass of these kinds of skills then cause symptoms to reduce. We also know that decentering is effective under acute stress. So if you take someone into a lab and you really stress them out, I don't do it, but I know a lot of researchers who have, it tends to be that if you teach someone how to decenter beforehand and you prompt them to do that in response to stress, it tends to really reduce their stress, reduce their distress. So how did we test this? How did we see if this is actually working, or whether this is something that could be helpful? So we did that with a randomized control trial. So how that works is we take a population, this is kind of young people, imagine all young people, and then we recruit a group of those to the trial. Then what we do is we split them into 2 groups randomly, and we put some of them in our treatment group, so our decentering group, and we put some of them into our control group. So the control group our kind of is them doing something else that's not kind of what we're focused on. There's lots of good things about doing this, but one of the kind of really good things is that it means that basically you expect the results to be the same no matter how you randomize people, kind of no matter what group you put them in. And also in our study, another good thing about it is that no one knew which group they were in. Now you might be thinking, Rachel, how did they not know what treatment group they're in? I'm about to tell you. So the way we did that is we made sure that our control group was as believable as possible. So our decentering group had a workbook that went along with the project, and we made sure that the control group also had a workbook. We had a nice little instruction video for our decentering group. We made sure the controls had that as well. And we tried to make sure that the control condition was as believable as possible. So in this, we were looking to test 2 things. So these are the kind of things you need to keep in mind. So the first is this proof of concept. So I know it sounds a little bit basic, but we wanted to check that if you put people in a decentering condition where they're learning how to decenter, do they get better at decentering? Which we were like, we just want to check that absolutely that's what's happening. And then we also wanted to check what the interaction was with mental health. So do people's mental health outcomes, do they depend on which group that they're in? So we did this using a kind of a small number of people. So this is kind of a first stage trial. And we recruited 114 individuals. So these were adolescents between 16 and 19. We had over 600 who who did a previous study to see if they would be eligible for this trial, and 547 of ours were eligible. The primary criterion for being eligible is that they scored 16 or above on a depression symptom scale. So what that means in practice is that they may not be experiencing kind of full depression, but they're struggling, or it seems like they're reporting that they're having kind of low-level symptoms. We were hoping to catch people who maybe are having kind of low-level symptoms, but maybe haven't got to a point where they would be accessing psychological services. So we then screened them more for other kind of eligibility criteria and randomized them either into our decentering group or our control group. So what were they doing in this intervention? So this was a 5-week intervention, and they had 5 days per week where they were doing things related to our intervention. So for our decentering condition, we kind of synthesized all exercises that were related to psychological decentering. from all schools of therapy, and we made these into 25 unique 10 to 20 minute podcasts. One thing I didn't think I'd be doing during the pandemic is making a sound booth in my closet and having my very patient audio engineer on the other end of Zoom trying to tell me how to set up a really fancy microphone that I wouldn't know how to use. And we recorded them all as podcasts. On Mondays and Tuesdays each week, they did a kind of broader mindfulness type exercise. The reason that we did that is because to decenter from difficult thoughts and feelings, you have to first notice that you're having difficult thoughts and feelings. You have to kind of be aware of your internal state, so we wanted to make sure that we were encouraging that. Then on Wednesdays, Thursdays, and Fridays, they were learning kind of practical decentering skills. So these varied depending on the week. And they were, so for example, one of the weeks it was kind of, they were thinking about difficult memory and then imagining it as if they were kind of a fly on the wall watching it. So seeing if they could think of anything different about it from that perspective. One of the weeks as well was something that I kind of mentioned before, which is thinking about a worry they had and then imagining what they think about that worry after kind of a month from now, a year from now. Would they remember it? What would they think about it? Those sorts of things. And all of these podcasts were available on Spotify, iTunes, kind of places where adolescents get podcasts. So our control condition involved another kind of set of recording for me as well, which was a lot to do, but was a lot of fun. So we had Mondays and Tuesdays. They had kind of light stretching videos, so kind of yoga type, like very, very easy stretching, kind of physical movement to match with the mindfulness, so that they were kind of doing something, but they weren't having any kind of internal focus to it. Then on Wednesdays, Thursdays, and Fridays, they were doing kind of cognitive training games, so things that were training things like kind of memory or kind of visual search. Things that we thought, because the decentering is it can be quite mentally tiring, we wanted to make sure that our control participants were also doing something that was matched for that. And they were spending about the same amount of time doing it. So that's what the control group were doing. And this is actually the first time I'm presenting results from this, so it's quite exciting for me. We weren't really sure if it was going to work, because we find that with interventions, it tends to be that doing anything is better than doing nothing. And we were wondering whether, especially the positive feedback we got from our participants about, they really enjoyed doing the movement videos, they really enjoyed doing the games. We wondered whether actually our decentering condition was going to do anything. So going back to the kind of questions that we were asking. So first one, does decentering improve with specific training? And the answer is yes. So here's another graph. So you've got decentering score on the left, going up, and then you've got the time point. We collected measures from our participants at the beginning of the trial, then in the middle, then at the end. And you can see the dark blue line, which is the decentering group, and then the control group, which is the light blue line. You can see that decentering did increase a little bit negligibly in the control group, but it increases a lot more in the active group, which we were obviously extremely pleased to see. Next question, does mental health improve with training or with kind of this decentering training? And the answer is also yes. So here's a couple of graphs. One of them is on depression symptoms at the top, and then you've got well-being down the bottom. So you can see as well that depression symptoms seem to kind of go along for a little bit up until the middle of the trial, and then they start to decrease. So we're kind of wondering whether that might be evidence for what we saw, kind of what we see with this kind of temporal effect, whether kind of learning decentering over time then makes symptoms start to decrease. And you can also see well-being increasing. One thing you might notice about these graphs is it looks like they have like a difference at baseline. So we control for that in our analysis, and yeah, so we make sure that that's not interfering with our results. So in conclusion, it seems like we can teach adolescents how to decenter, and it does seem like this improves their mental health. Of course, this is like really early stage research, so I'd want to do this on a much bigger sample. Our participants were paid to take part, so we'd want to try and do it maybe with participants who weren't being paid, because then it's probably not going to be affordable. But yeah, there's a lot more that needs to be done. But yeah, obviously we're very excited to present these results. I just want to thank you all so much for coming, and that little scan me there will take you to a Google Drive which has all of the podcasts on there, so if you want to have a little listen, you can. And I just want to say thank you as well to my team, my funders, my supervisors, all my participants who made all of this possible. It was really a huge job, and I couldn't have done it without any of them. I welcome your questions.
Could do with some of that myself. Good. So, questions for Rachel.
Bob.
So Rachel, how do the improvements compare with the expensive treatments?
Oh, so we haven't done so one of the things that we would do if we were doing a larger trial is do some kind of health economics outcomes. So we've done that on kind of larger projects in our group. We haven't started doing that yet. So yeah, we don't yet know.
I'm thinking that there's presumably published data on the effect of in-person therapy, for example, on the same measures as you have.
Yeah.
So is this like sort of same size of things you see in published papers saying, hey, our therapy works?
So I think what we do know is, so we look at something called effect sizes, which is kind of looking at how much a treatment really, how much it's really making a difference to our participants. And for a low intensity intervention, you would expect an effect size of about 0.3, and we had effect sizes of 0.7, so ours were quite large for something like this, where you're basically giving someone a load of podcasts and saying, listen to this every day. It's kind of very, very kind of low cost, self-guided. That was actually quite big. That accounts for, so what that means in real scores is that would be about a 4-point decrease on our depression scale. So that, I think, in our study, it brought about 10% of our participants out of the range of at risk and into the healthy range. Does that answer your question a little bit better? Yeah, perfect. Hi.
How do you think this would apply to adults suffering from depression and anxiety and the like? Is this scale beyond teenage years, even in teenagers, quite critical to address mental health?
Yeah, for sure. Yeah, I think I can't see any reason why we wouldn't want to kind of scale it for adults. I think, yeah, we tried to kind of, with the podcast and the language, we tried to kind of make it, well, we wanted to make sure that it wasn't kind of patronizing, so it wasn't kind of like pitched too young for our participants who were kind of 16 to 19. But yeah, I think we maybe would kind of re-record for adults, or that, to be honest, I think maybe adults could use the same ones. Yeah, I don't see any reason why not. Well, why we wouldn't be able to use it for that.
Yes.
In your graph, you had a graph of your people you recruited and you were showing the cut-off.
Mm-hmm. Mm-hmm.
Were they randomly selected? A huge majority had a high score.
Yeah.
Yeah.
That's something that I've been thinking about a lot. So these participants were taken from, this was, anyone could kind of volunteer for that project, anyone who was in the sixth forms that we applied to. So what you do find sometimes with psychology studies is maybe people who have more of an awareness of psychological symptoms self-select. So it could be a selection bias. However, it is kind of a bit in line with what we've been seeing, which is that especially kind of in light of the pandemic, there are a lot of adolescents who are identifying as having kind of struggles with low moods. So it's, yeah, you're right, it is a huge amount, and it was more than I thought would qualify, which in some way made the job of picking people to be part of the trial easier, but of course is a bit concerning. I completely agree.
Still a fairly normal distribution, whereas you'd expected what you say is true for it to be skewed like
Yeah.
some.
Mm-hmm. Yeah. It's something I think about a lot. Yeah.
Great. I think we should move on. But thank you very much, Rachel.
Thank you so much.
Very good.
Okay, so somebody help me.
Our next speaker is Bob, who's going to tell us about how you hear with a bionic ear.
I wish I'd used that title.
I can't promise a rhyme on every intro.
No. Okay. So thanks very much for coming, everyone. My name's Bob Carline. I'm one of the deputy directors here, and I run a research programme on listening through a cochlear implant. So in order to explain how that works, what I want to do is quickly run you through what happens in normal hearing. So we can see here the sounds coming in on the left of the... I'll just make this a little laser pointer. How about that? Seamless. Coming in on the left here. It's coming into it's captured by your outer ear. And this causes the eardrum, which is about the bit which, if you stick your finger in your ear, that's about as far as you'll get to vibrate. This makes some little bones in the middle ear also vibrate, and they transmit information to the cochlea, which is this spiral-shaped organ here. And along the length of this cochlea, there's a membrane called the basilar membrane, and it's thin and stiff at one end, at this end, and wide and wobbly at the other end, which is called the apex. And what this means is that low-frequency sounds tend to excite the apical part of the cochlea, where the membrane's wide and wobbly, and high-frequency sounds activate this bit here, which is called the base. And so each part of the cochlea is actually tuned to a different frequency. And along the length of this membrane, there are some sensory cells which transform that vibrations into neural signals, which are then conveyed by these nerve fibers, which are sort of coming up into the middle of this spiral. And each of these nerve fibers then conveys information to the brain. So each one of these nerve fibers will be transmitting information about a particular frequency component of the incoming sound. Now, if you have, like me, a bit of a hearing loss, what happens is that some of your receptor cells die, and you might get a hearing aid. I should really have one. I keep meaning to get around to it. Not a very good advert for my own research. But if all of my hair cells died, then I wouldn't be able to hear at all, and that would be quite devastating for me, both socially and professionally. And so, in that case, I would get a cochlear implant. And what cochlear implants do is to restore hearing to these very, very severely and profoundly deaf people by bypassing the damaged hair cells and directly stimulating the auditory nerve with trains of electrical impulses. And as a result, quite miraculously, many patients are able to then again understand speech pretty well, at least in quiet. But unfortunately, there's quite a lot of variability. Some people do well, but there's a minority of patients who do quite poorly, and even the most successful struggle in noisy situations. And these implants are now available in the UK on the NHS, from babies down to about 4 months. 92 years is the oldest I've heard of, but I'm sure that record is increasing. So here's how it works. If you look at this part here, this is a microphone behind the ear. It looks a bit like a hearing aid. And it's connected to a speech processor, which is essentially determining what the pattern of electrical stimulation is going to be for each of the electrodes in the cochlea. That information is transmitted across the skull using a radio frequency transmitter, which has got a little magnet in it. And inside the skull, there's a receiver, which has also got a magnet, which allows you to hold it in place. And then, in fact, one of our younger patients likes to hold her party trick is to hold a fridge magnet to it at parties. You know, okay. Surgeons don't like to know that. This receiver transmitter then, stimulator then figures out what the exact pattern of electrical stimulation is going to be and transmits that information to an array of electrode inside this tiny snail-shaped organ, the cochlea, which I showed you earlier. And here's an example of an implant type with 8 electrodes. And again, you can see there's the low frequencies at the apex and the high frequencies at the base. And these little whiskers you can see are the auditory nerve fibers coming up through the middle of the cochlea. And they're the bits which would normally synapse on the basilar membrane, but here we're directly stimulating them with electrical impulses. And if you look at the response to a sound such as the vowel R, and we just take one of these electrodes, what we can see is that it's stimulating the nerve with a train of quite high frequency pulses, but there's some modulations in there. And in this case, these modulations are in time with the speaker's vocal cord vibrations, and it's conveying information about the pitch of the sound. So these fluctuations are going along roughly at the fundamental frequency of the sound. One of the things we've been trying to do is to see if we can improve upon these stimulation patterns. And what we thought we could do is the modulations I just showed you, I thought we could remove them, enhance them if we remove some of the low amplitude pulses, which we think would have been masked by the preceding pulses. So here's again this example I showed you of the vowel R with the standard strategy, and here's the word the. And in our new strategy, which we call TIPS, Temporal Integrated Processing Strategy, you can see that we've basically removed lots of these pulses in the valleys, and that's enhancing these modulations in the vowel, and it's also enhancing some of the information we're getting about the thus, and it's making some of these onsets, for example, this offset is a bit more abrupt than here. So what we've done here is to study this in a speech and noise task, and what we're measuring here is essentially how loud you have to make the speech sound in order for you to be able to hear it against the background noise. So in this graph, down is good, happy face, up is bad. Here's the standard strategy, and we can see we're getting about a 2.5 decibel improvement, which is reasonably good. It's pretty cool. So the other thing which we have as an advantage about this is that it's saving battery power. When I talk to cochlear implant companies about this, I say, hooray, we're improving speech perception, and by the way, we save battery power. And they go, you can save battery power, and you're not making things worse? So they really want to save battery power so they can make their devices smaller and lighter and easier to use and have more processing in them. So we're all kind of got slightly different priorities, I guess, but we're all aiming to try and make life better for cochlear implant patients. Another thing we can do is to sort of drill down into exactly what's happening at the very auditory periphery. So again, here's our electrodes with our healthy neurons. And there are really two things which can sort of go wrong, if you like, which can make some electrodes not convey information very effectively. So one of them is the health of the auditory nerve. So here we got this healthy nerve, which I've shown you here. But we know that as people get deaf and get deaf for longer and longer, these nerves die away, and they can do so in quite a patchy way. So rather than being healthy like this, we can get some dead regions, which I'm shown in grey, where the nerve fibres have disappeared. And you can imagine it's probably not going to be a good idea to stimulate an electrode which is in the middle of one of these dead regions. And the other thing is that the cochlea is full of fluid, which can make the current spread quite widely. If you've ever been unwise enough to drop a hairdryer into a bath, you'll realise fluid tends to conduct electricity, not very good. And so although we might be lucky and get one electrode here which produces a narrow current spread, which is what we'd really like to do, so that the frequency bits of sound, bits of the frequency region of sound which we're transmitting with this electrode, just stimulate neurons near that and nowhere else. We might get another electrode which has got a broad spread, and that can make the sound appear a bit blurred. And worse still, we might get something, with, for example, with a broad spread which is in a neural dead region. And the way in which we're trying to unpick all this is that quite a cool thing about cochlear implants is you can not only use these electrodes to stimulate the nerve, but you can also use them to record neural activity. So we can stimulate with one electrode and record the resulting neural activity from another. So we can do that for every electrode. And what we can also do is precede each electrode, a pulse on each electrode, with a pulse on some other electrode, which will basically tire the neurons out and see how much they interfere. And you can imagine that for this electrode here, everything would be fine. I could stimulate another electrode over here, and it's not really going to have any interference because it's going to stimulate a different set of neurons. But if everything's a bit blurred, we've got broad current spread, then we might find interference from a wider range of neurons. And something different still is happening up here, because here, this electrode is actually stimulating neurons not really close to it, but close to some of its neighbours. And so what that would mean is if I stimulated its neighbours, that would have like a really, really big interfering effect. Whereas if I stimulated something over here in another dead region, it wouldn't have any effect. So the reason we're doing this is that we have a clinic at Addenbrooke's Hospital with some of our colleagues where we have patients who are having particular difficulty with their implant. And what we'd like to do is, assuming we have a patient who's got some neural survival pattern a bit like this with some dead regions, what might be happening is that some of these electrodes are producing really broad spread and some of them are producing narrow spread. Some of them are in a dead region, some of them are in a healthy region, and you're just getting a bit of a mess. And what we want to be able to do is to turn off some of the electrodes. This might be a case of less is more and produce a cleaner representation of speech. Finally, I'd just like you to think about what would happen if you were deaf for several years and suddenly you got this operation, you got a cochlear implant. A few weeks later, you go to the audiologist and they activate your sound, and you can hear, perhaps for the first time, properly in months or years. And some things are going to change over time. So one of them is that speech will sound really quite unusual and also be quite hard to understand. So your scores on a speech test are going to be quite low. And one of the things which happens in the months following implantation is that people get used to the sound and their speech scores improve. And that's partly due to learning. We know that people get to be pretty good at mapping this strange new sensation into their memory of what words are like or what speech sounds are like. But we also think there are some sensory things going on in the brain which aren't just learning. And one of the things we know about is that each time you will go back to the audiologist to get your implant adjusted, they're going to have to turn the juice up more. Basically, the sounds are getting quieter, and some patients we have, it's an excess, we call them power junkies. But essentially, the sound level's got to keep going higher and higher and higher. And we're investigating what's the neural basis of some of these changes. And so far, what we've been looking at is this change in loudness. And what we've been able to do is to use EEG to try and make recordings of neural activity at these different stages of the auditory system. Actually, from the auditory nerve, we can just use the recordings from the implant itself, like I showed you earlier. But these are measures in the brainstem and the thalamus and the auditory cortex. And we can distinguish these measurements because as we move further up the system, the responses occur later and later. And so what we can then do is as a person's loudness percept changes over time, we can map that to changes in the responses at all these different stages to try and find out which parts of the system are responsible. It may well turn out to be that this differs across patients. So I'd just like to summarize. This is a small sentence here. It's cochlear implants restore hearing to deaf people, but it's actually massively important. Every time I say that, I think, gosh, you've got a bionic ear. And it works. It's hugely successful overall, but we have varying outcomes. We have a processing strategy which saves power and improves speech perception. We can record from the implant to try and work out on a patient-by-patient basis what's actually going on right at the level where the implant starts interacting with their brain for the first stage and the level of the auditory nerve. And as well as the clinical benefits, it actually provides a natural experiment in plasticity in adulthood. There's not really many other sensory modalities where you can lose that modality and then where there are a large number of people who suddenly get it back again all of a sudden. So I'd just like to finish by thanking my colleagues who are involved in this research. It's probably not a coincidence that I'm the oldest person speaking here and I have more people to thank in my talk and, of course, my funders. Thank you.
Great. Questions for Bob? Yes.
Yeah.
You're recording how people's brains change to in response to having the implant and adapted to it.
Yeah.
My first thought is, wouldn't the implant actually interfere with most of the techniques for measuring brain? Obviously, you can't stick someone with metal in the head in an MRI.
No.
The signals might, I don't know if they would, but I assume that if you're using electrical signals, they might interfere with the EEG.
Yeah,
so how do you actually achieve that?
So it depends on the measure. That is absolutely the point. Electrical artifact is to a cochlear implant researcher like headwinds are to a pilot. So for some of these peripheral things, you can just present pulses to a relatively slow rate and measure the responses in the gaps between the pulses. For other stimuli, we might, for example, use amplitude modulated pulse trains, a bit like we have in the implant. And essentially, we've developed with colleagues a custom-made EEG system, which has got a 260 kilohertz sampling rate, so super, super, super fast, which is fast enough to be able to sample these artifacts, and then we essentially blank them and linearly interpolate between them. And we have a few other techniques, for example, where we put in two stimuli at different frequencies, and then we know the brain is nonlinear, and the brain actually introduces a component at another frequency, at a distortion product frequency, at the difference between them. And that's quite nice, because we can then measure that, and there isn't any energy at that frequency in the artifact, but we can just look at this distortion product. So it's a big part of what we do is hunting down these electrical artifacts and getting rid of them as much as we can. But that's an excellent question, because it really is. And it's one of the things we're really fortunate to be able to do. We've got some great collaborators around the world and some really top people working in-house on that.
Yeah, I think Bob is about to take you on in his research group. Thanks for that question.
Yeah.
Other questions for Bob? Yeah.
One of the pictures you showed, the
cochlear implant appear to be on one side of the head. Is that just the receiver, or do you only actually implant in one ear?
So in the UK, if you're an adult and you're deaf on the NHS, you only get an implant in one ear. In countries with private medicine, you'll often be, it's better to have an implant in two ears. If you're a child, you can get an implant in both ears, provided you have them done at the same time, because that's a bit cheaper. And if you're an adult and you have some other disability, for example, if you're blind, then you're allowed to get two implants. So it is definitely better to have implants in two ears.
So if there are different dead regions in different ears of cochlear, you could optimise the different ears.
That would be a cool, yeah, that would be a cool thing to do, and I bet the brain will be able to figure it out if you have big enough dead regions. But also, it's just it gives you a sense of localization, which is what's known as the better ear effect. So if somebody's talking to me here and my implant's on this side, and there's somebody annoying over there, then that's not so good. So it allows you, in normal hearing, we can switch which ear we attend to, and cochlear implant patients can get that benefit too.
Great, good. Let's thank you very much, Bob. And we're now going to do some language. So Abby's going to tell us about some of her stuff. Let me get it. Slides. Here we go. Over to you, Abby.
Excellent, thanks. So we're staying with a similar theme actually, so I also do research on speech. But the focus of my research is not on what we say, but the way that we say it. So generation of a voice requires the precise coordination of many different muscles, as I'm showing you in this video here. And if we think about this in the context of movement in general, professional dancers train for many years to be able to perform complicated sequences of movement. And yet here I am, as I'm speaking to you, performing this sort of internal dance routine, if you like, in order to generate my voice. Now, the precise sound of our voice is shaped by a variety of different factors, some more stable to do with our physiology, like our body size and our gender, which affect voice pitch, but also some more dynamic to do with the context in which we've learnt to speak, so the region in which we grew up or what language we grew up speaking might give us a particular accent. Also, more dynamic factors like our state of health or our emotional state can affect how our voice sounds. And also, the sound of our voice can also convey a lot of sort of social identity information about us. So altogether, there's a huge amount of variability in how different speakers produce speech sounds, even within the same language, such that no two voices are ever the same. And I want to convince you this evening that this actually represents something of a challenge for the brain. We really take for granted the fact that the brain is able to take this highly variable input from lots of different voices and map it onto a shared underlying understanding of what speech sounds form particular words. So to take an extreme example, if I was asked to name this red fruit, I would say that's a tomato, but a speaker of US English would say that's a tomato. Now, in this case, the brain has to recognize that although these two sound different, they do in fact correspond to the same word. A perhaps more subtle distinction closer to home would be the distinction between Bath and Bath across different regions of the UK. But I want to further convince you that even when the brain is faced with two speakers producing the same pronunciation of a word, so Bath and Bath, the precise sound of their voices will still be very different, and the way in which they produce the speech sounds will be different according to differences in the physiology of their vocal apparatus and just the particular way in which they've learnt to say those speech sounds. On the production side, we know that the more that we're exposed to a particular pronunciation of a word, the more likely we are to produce that ourselves. So let's say we move to a different part of the UK and suddenly we hear lots of people saying it as Bath. If we've, the idea here would be that we've maybe started out with a target for how we think this word should sound, that might be Bath, but through exposure to these alternative pronunciations, we update our target and start producing this ourselves. These more obvious changes in pronunciation might develop across sort of longer timescales, but there's also evidence that even within a single conversation, the voices of two speakers can start to sound more similar to one another on a range of features like the pitch of their voices, the intonation they use, their speaking rate, and even some aspects of accent. And I think some of us may be more aware that we do this than others if you've ever found yourself feeling like you're picking up aspects of someone else's accent. And of course, there are limits to this process, so it's not the case that we come away with a completely different accent each time we talk to a different speaker. And so the brain therefore needs to balance, on the one hand, the need to maintain a consistent vocal identity, so with stable internal targets for how we think particular words should sound. But on the other hand, it's important that there's a certain degree of flexibility in these targets so that they can be updated when we're exposed to other voices. And so my research looks at the interplay between these two and how they determine the sound of our voice. So to look at this, I typically do the following type of experiment. So I get participants come into the lab and I give them a headset microphone and a pair of headphones, and I ask them to read aloud single words, like the word bed. And I take the signal from the microphone and send it to a computer, and that computer makes a slight change to the sound of that speaker's voice. So to illustrate how this looks, let's again go back to this idea of kind of targets for speech. So the center of this target is the participant's target for the word bed, so they're sounding like this.
Bed.
And the change that the computer makes is to make the F vowel sound closer to the A vowel sound, as in the word bad. So it sounds something like this.
Bad.
So that's sort of somewhere between bed and bad, hopefully you can hear. And I take that altered signal, I send it back to the participant through the headphones in real time, so that when they're saying the word bed, what they're actually hearing themselves say sounds closer to the word bad. And this has the effect of essentially pulling them off target and creating a sort of error between how they expect to sound and how they're actually sounding. And what we find is that speakers will unconsciously compensate for this by changing the way in which they produce the speech sound to move in an opposite direction, as shown in the purple arrow. So what they end up producing is something closer to the word bid, so something like this. Oh, sorry.
Bed.
In order to sort of counteract the effect of the error that we've introduced. And what this shows us is that kind of internal dance routine that I started my talk with doesn't unfold in a rigid manner, but is actually being continually adjusted according to what the brain hears as we're speaking. Now, most of this research has just been done while speakers are producing speech on their own, but in my work, I've been trying to take this into more interactive speaking contexts where there's also another voice. And in particular, one useful task that I've been using for this is synchronous speech, which is the act of speaking in unison with another talker. So in this setup, I typically get participants to read full sentences, but at the same time, they're hearing another voice through the headphones saying the same sentence, and they just have to coordinate the timing of their speech to be in synchrony with that other voice. What this means is that the brain isn't just hearing the self voice, but also another voice, and these two signals will be very overlapping in time. So the job of the brain is to separate back out these two signals. We know that it's using the sound of the self voice to make sure, for monitoring, to make sure that we stay on target and correct for any errors. But what about the other voice? How might the brain respond to that? So in an initial study, I looked at this in the context of voice pitch. So I had speakers read a series of sentences, firstly on their own, to get a measure of their baseline pitch, as shown in the blue X. And then I asked them to read the sentences again in synchrony with another voice. And this voice had been manipulated to have quite a high pitch, so represented by the green X. And what I found was that during the synchronous speech task, participants tended to increase the pitch of their voice in order to sort of converge to or imitate the other voice. And this was all unconscious, so there was no explicit instruction to do this. And I want to just highlight at this point, therefore, that there seems to be a difference in the way that the brain is responding to a self voice versus another voice. So if we go back to the first type of experiment that I introduced to you to, where we're introducing a change in the sound of the speaker's own voice, the brain responds by moving us in an opposite direction to try and counteract the effect of that change. However, if instead the brain recognizes it as someone else's voice, it responds by moving us towards that other voice, so that we imitate it and converge towards it. And I was interested then in what happens if we try and look at these two responses in the same experiment, so which is what happens obviously in natural speech. We hear our own voice and we hear other voices as we're talking with them. And what happens explicitly if we pit these two responses against each other. So to do that, I combine these two experiments. So people are reading sentences and having the sound of their own voice changed, as in the first type of experiment. So we'd expect them to move in the direction of the purple arrow. But at the same time, they're synchronizing their speech with another voice, and that other voice is over in this direction, shown by the green X, relative to the participant's own voice. And so in this case, in order to converge to the other voice, they actually need to move in the other direction, shown by the green arrow. And so we've introduced a conflict between where the self voice is pulling them and where the other voice is pulling them. And I was interested in, might one of these responses maybe dominate over the other? Might the self voice be prioritized? Or do they just cancel each other out, essentially, to result in no change? And surprisingly, we actually found the latter was the case. So people in this task show very few changes in the way that they're producing speech sounds, suggesting that the self voice and other voices are sort of equally weighted in determining the sound of our voice. And just to show you the kind of reverse condition, another group who did exactly the same, but this time the sound of the other voice was manipulated to pull them in this direction, so in the same direction as the self voice. And in this case, we saw very large changes in the expected direction. So it seems that these two responses are just sort of summing up together. So I hope that I've shown you this evening that as we're talking, the brain is really sensitive to what it hears, but it really matters who is talking. So the brain appears to do different things with what it hears, according to if it's recognized as a self voice or someone else's voice. And this means it's really important that the brain can distinguish between those two things. And it's thought that a breakdown in this process might actually underlie things like the experience of hearing voices in schizophrenia. So the next steps for me during my research here at the unit is to take these type of experiments into an fMRI scanner and look at how they manifest in activity patterns in the brain. So I'd like to say thank you very much for listening, and I'd be happy to take any questions.
Very nice. Questions for Abby? Yes.
Very interesting. I was wondering if you thought the same behaviour would happen for someone that was maybe bilingual in their second language, or if it would happen, for example, with someone that's got a cochlear implant and their signals maybe a bit different.
Yes, those are both excellent questions. So there's actually a PhD student working within my group looking at this in bilinguals and also cross-language differences. So there does seem to be some cross-language differences. I think in English we see these effects very robustly. In some other languages where the vowel sounds, like in Spanish, are very far away from each other, you don't see these sort of, you don't see as much correction for the same amount of change because it's not pushing them into another vowel category, if you like. But also she's been finding some evidence that you do actually see cross-language transfer of these effects. So if you are speaking with an altered sound of your voice in English and then you speak in your second language, you do actually show transfer of that learning into the second language, which is interesting and perhaps not what we'd expect. In terms of cochlear implant users, this is something I am very interested in. The sound of speech, and this is an area that I'm kind of just learning, so I'm sure Bob will correct me on things, but the sound of speech through a cochlear implant strips it of a lot of what we'd call it spectral detail. So a lot of information on the types of things that we're changing to change the sound of a vowel. A lot of that is quite reduced and represented quite crudely. So I think it's unknown to what extent cochlear implant users would be sensitive to these sort of more fine-grained changes to the spectral content of the speech. But that's something that I'd be really interested in researching. Yeah.
Yes.
These different effects as a function of the source of the speaker are very interesting, and I'm curious whether that source identification, source processing underlies the experience of
listening to our own voice in a recording.
Yes.
It's horrible. It's so awful. Part of that is because the experience of hearing our voice as we're producing it, we don't just perceive our voice coming through the air as we do other people's voices, but we also receive what we call bone-conducted feedback. So this is actually sound waves traveling through our bones. And that means that the sound of our voice is quite different when we're actively producing it compared to when we're just listening to it, because there we don't have the bone conducted feedback. So that's partly why, explains why it sounds so different, yeah. Thanks.
Yeah.
Hey.
Thank you, really interesting talk. You described two different mechanisms for self-speaking, hearing yourself and hearing someone else. The effect went in the opposite direction.
Yeah.
Has anyone played with ambiguity of whether the source of the speech is you or someone else? And would
Yeah.
would you expect that would just disrupt the entire mechanism? Or could
Yeah.
be some sort of sum of the two? I don't know,
Yeah.
effect direction.
Yeah, that's a really interesting question. So there has been some work looking at, because obviously at some point when you change a person's voice so much, maybe there'll be a point at which they stop recognizing it as their own voice and recognize it as someone else's voice. But actually there's been a lot of interesting work showing that if you play back someone else's voice, but in a way that is contiguous with your own voice, then actually people do start to take ownership of that other voice, because it's sort of temporally correlated with your own speech productions. So we call it the rubber voice illusion. So you kind of take on ownership of that other voice, and actually there's been some really interesting work looking at what happens when you've had experience with speaking with that different voice. If you then change the sound of that voice, people do show these similar kinds of opposing responses where they're correcting for changes, even though obviously globally the sound of that voice is very different to that actual voice. People do seem to take ownership of it and then be sensitive to these sort of smaller changes. So I think actually our sense of agency of a voice is quite more flexible than you might think, which makes sense because the sound of our voice changes massively across the lifespan. I mean, when we go through puberty, there are lots of changes, so it makes sense that this would be adaptable, I guess. But yeah, really interesting. Thanks.
Yeah, thanks, Charlotte. So, shall we say thank you to all the speakers again? And thank you again all for coming. I hope you've enjoyed it and found some new things and some things to follow up. It's been great to have you here, and a safe journeys back to wherever you're going. I can hear something outside, so I don't know if that's the rain or